So I'm Matt Hashimoto.
I'm Cameron.
I'm Kyle.
And we decided to do our research on ALS observation and diagnosis.
So the main topic that we focused on is how can we better diagnose ALS at earlier stages? So ALS, or amyotrophic lateral sclerosis, is a very elusive and complex degenerative brain disorder. So scientists right now, they currently don't know much about what causes it. They have some theories about it's either protein, genome-based, but they're not really sure. So our approach is to not find a cure for ALS, but to find a way to diagnose ALS earlier. So the way we are going to do it is a protein released by a motor neuron when it dies is called a neurofilament medium polypeptide, or NEFM for short. So when it is released, we can examine it from people with ALS and people without ALS, and we can further examine it with using a fluorescent antibody tagging system and a light sheet microscopy. And if we are able to figure out how to continue with this research, we could save thousands of lives across the country.
Okay, and now on to precedents. So in current technology, scientists possess many methods to which they can image the brain and produce models and examine it to any extent. But it is a little bit limited as of now. That's why we have the Brain Initiative. And so some of these techniques are fMRI, CAT scans, and then they can also image dead samples. And let's see. So next, there are stem cells. So stem cell research is hot and new right now. It's been progressing over the last few years. And although they haven't advanced it enough yet to start using it to its full potential, but hopefully we'll see that in the future some near time. Still under stem cells are iPSCs, or induced pluripotent stem cells, which scientists can use to grow any type of cell in the culture and study it there. And lastly is protein analysis, which is the study of proteins in CSF and blood, which can be used to diagnose ALS, and I'll get into that more later. And then so on to our project proposal. So we propose a method for diagnosing ALS using a protein normally found in motor neurons, which he talked about earlier, which is called NEFM, or neurofilament medium polypeptide. And that protein plays an important role in intracellular transport, as well as just general structural integrity of the neuron. And so in ALS patients, what they've observed is that in their CSF and their blood plasma, there is an increased level of these proteins. And so the first step is to find willing volunteers with and without ALS for spinal taps. And to do that, we must take multiple samples. And then the second step is to tag the protein with an antibody. So the way this works is a two-antibody system. So one antibody binds to the protein first, and then a second fluorescent antibody binds to that first antibody. And so what that does is it makes it easy for the protein to be imaged with fluorescent microscopy. And we will use LSM, or light sheet microscopy, to accomplish this. And in the disease, we should see something like this on the slide here, is lots of fluorescent dots, and those are the proteins. And then normal patients, we should see little to no fluorescence. And then finally, the third step is to determine if the NEFM protein is the same in normal patients and in ALS patients. And this, we can either isolate the protein from the CSF and examine it, or we can take a biopsy of motor neurons.
And then about the social context. By researching a new way to diagnose ALS earlier, many lives can be saved. By researching a diagnosis, it brings us a step close to understanding how ALS works and develops. With an earlier diagnosis, it may be easier to study the early development and stage of ALS. This may be able to provide the people living with ALS in the United States alone. Approximately 20,000 to 30,000 people have ALS, and 5,000 more are diagnosed every year. With the research that will be done, a cure or diagnosis indicator that will drastically help the society. Then about the challenge. One challenge may be finding willing volunteers for spinal taps. It's not a very fun procedure. To say the least, we may also have to try to obtain as a random sample as possible to preserve statistical accuracy. The test is not 100% for ALS, as other diseases may cause the death of neurons, and then, in turn, an increased concentration of NEFM.
All right, so future directions. So this diagnosis will take us one step closer to either finding a cure for ALS or finding a more complete diagnosis. So using our technique, we can use high-resolution imaging, such as light sheet microscopy or maybe even transmission electron microscopy, to further study a cure for ALS. So for example, if it turns out that it is a genetic modulation in the ALS cells that will cause it, we could, new biotechnologies such as maybe CRISPR-Cas9, which is a kind of protein that has been studied to alter genetically some stuff. If we can apply it onto this concept, we may be able to alter the genes of ALS symptoms and then maybe fix the structure. So in essence, we want to create a new type of early diagnosis for ALS. So we will use the protein NEFM, which is released during the motor neuron's death. So when motor neurons die, it's usually a symptom of ALS. So we hypothesize that elevated levels of this protein in the blood and cerebrospinal fluid can indicate that the sample may have ALS symptoms. So this, with the motor neurons dying so frequently, this can lead to major brain damage and muscle dystrophy. But muscle dystrophy is basically when the muscles shrivel up and become very weak. So we will use LSM and fluorescent antibody targeting systems to track them. So in conclusion, when we see lots of NEFM protein in the blood and cerebrospinal fluid, we can then conclude that it's most likely that there will be symptoms of ALS. So we will want to use this in the future to be able to possibly diagnose ALS earlier before major symptoms start to occur.
Thank you. Then here are contributions.
Okay, so light sheet microscopy, we can use it to study live cells, which is always helpful because the light is minimally invasive onto it. So if you, in a traditional confocal microscope, if you were to use laser, it would take like pixel by pixel, right? Go like this. But in the laser, it can go straight, so it reduces the amount of toxicity which will be exposed to the live sample. So in our case, if we take the sample from the spinal tap and compare the two from someone with ALS and someone without ALS, then we could examine the levels of the protein that we previously tagged with the antibody and see, like, if it's consistent, the data is consistent, we could hypothesize that if there's elevated levels of the protein, then it could lead to more likelihood of ALS.
So I need to clarify, or need you to clarify. So you said that this NEFM is secreted as neurons die, right?
Yeah.
So and it's present in cerebrospinal fluid and in blood.
Yes.
So it's not in the cells anymore, right?
No, because so the protein is helped in structural. So when the neuron dies, the protein is a polymer. So when it dies, it breaks apart into the individual monomers and can spread out throughout the system.
Okay, so if it's then not encapsulated inside the cell anymore, then with respect to the last question, why does it matter to use light sheet, right? Because you're not trying, the cells are dead.
Yeah.
And the protein's not in the cell anymore. You're just trying to detect the presence of the protein in the fluid, right?
Yes.
So in that case, does the light sheet add anything for you?
We could, light sheet microscopy is very useful in using 3D renderings as well. So you could take it layer by layer to examine the shape of the protein and see if there's anything different between the two.
Another simple question. Do you know what the different level of concentration, let's say a patient has ALS, right? Everyone keeps saying no one wants a spinal tap. I agree,
Yeah.
right? So what's the relative level of this NEFM in like serum versus cerebral spinal fluid? Like, if it's in the serum or in the plasma, why is there just so little of it that you can't use that instead of CFS?
Well, let's see. So for cerebral spinal fluid, I guess is, I think it would be easier to maybe see the proteins in CSF. I'm not sure how to really explain it, but.
So like research is very like experimental right now, so we don't know much more about it because there's lots of complications and variables that will change the results.
I mean, it's most likely that the concentration of the protein is higher in the spinal fluid because this is where the source is. But it's all, as you mentioned, it's probably also going into further grades, maybe with the blood. This is maybe a place where you can also like sample it and have an early diagnosis, right? So you can imagine you doing both, right?
One more question. Next question back here.
Besides ALS, are there any other brain diseases that also release this protein, and how you differentiate and have systemic comparison?
So like Cameron said, this kind of testing is just like one of the first steps in analyzing, because other brain disorders, when they degenerate neurons, they will most likely release the same protein. So then we can at least see that, oh, something's wrong here, so we need to do further testing.
That's my question. How do you know it's ALS versus the other brain disease?
And
Right now, we are not sure if it can. We know that we can use this protein to distinguish that there is something wrong. It's just ALS is one of the major applications that this could be found in, and that studies have shown.
All right. Thank you, guys.
okay.